All right, so what we're going to do is talk about algal ecology, and we're going to start by reviewing the 6 functional groups of phytoplankton, 6 functional groups of algae in lakes, and then look at them from some different perspectives. Under what kind of conditions do you get lots of blue-green algae? What kind of conditions do you get diatoms? And why? We got all these different types of algae that live in lakes, and yet lakes are really, it's just water. It's actually a mystery why there's so many different species of algae coexisting. In fact, it's called the paradox of the plankton. It's a famous mystery in ecology, why there should be so many different species of little green things that all, apparently live in the same place. And if you want to use the word niche or niche, they all seem to occupy the same one. So how is that even possible? Really kind of makes you realize that word niche or niche is not actually all that meaningful, because as far as we can tell, it just looks like there's one niche out there, algae. Why should there be hundreds, thousands of different species, even coexisting in the same lake at the same time? So, there's a lot of mysteries about algae, and one of the things we want to think about is even if we group algae into major categories, like these functional groups, some lakes will have lots of one of these groups, and a different lake will have lots of others. Some lakes will be diatom lakes. Some lakes will be filled with blue-green algae. 2 lakes that really look pretty much the same. One of them will be filled with blue-green algae in the summer, and another one might be filled with diatoms or dinoflagellates in the summer. Of course, people care about dinoflagellates because they cause red tides. And some dinoflagellates, they're like aliens from some sort of science fiction movie, make toxins, and they have like weird life cycles where they're like amoebas, and some of them are, really amazing. They live symbiotically inside coral, inside animals. Why are there dinoflagellates who are such like amazingly powerful, crazy creatures really abundant in some lakes, but not most others? And of course, people care about the cyanobacteria because one genus, Microcystis, has a species, Microcystis aeruginosa, that produces a toxin, microcystin. And that's fine, unless you kill it, in which case the microcystin is released into the water, and it can make you sick. Or, and every now and then, maybe even every year, a dog somewhere in Minnesota dies from drinking water contaminated with microcystin. And you might think, well, 1 dog a year. There are far more dogs every year killed by all kinds of other things, like cars, for example. And we don't talk about banning cars just because of this. But if there's something in the water that can kill a dog, then maybe there's something in the water that can hurt humans. And if there's something in the water that can hurt humans, oh my God, you bet every single water treatment plant in the state is going to care about that, because you don't want to be in charge of St. Paul drinking water and have something potentially toxic in your water. You don't want to be in charge of the drinking water, even in a tiny little city somewhere out there. So, we care. Why is it that one species or one genus or one group of algae dominate one lake and not another? Why at one time versus another? So that's algal ecology. That's the real question. Why are there more of this in one time or place compared to another time or place? And that's the fundamental, the central question of ecology, I think. Why Why is there more of one species at this time compared to a different time? Why is there more of this species or genus or family in one place versus another, one lake versus a different lake? Well, there are other questions that ecologists address. That, I think, is just the central, fundamental, most historically central question of ecology. But there's other questions like, why do some lakes produce more biomass of algae than others? Why do some lakes produce more biomass of fish than others? Why do some lakes produce more biomass of the top carnivores, you know, the game fish at the top of the eutrophic pyramid? What is it that controls the abundance of things? What is it that controls the production of things? Productivity meaning the production of organic matter. Some lakes, they might have a lot of algae in them, but the algae's not growing. It's just hanging out there. Other lakes might not have that much algae at any moment, but the algae's growing really rapidly, constantly making more algae. So what is it that controls productivity, and how is that compared to biomass? The mass of the algae versus the production, the rate at which mass is produced. What is it that controls that? And why do we care? Why do we care? Because, well, of course, if a lake has high productivity of phytoplankton and you harvest all the phytoplankton to make the lake clean, and then, you know, 2 weeks later, there's just as much as there was before, very high productivity. Or maybe what you care about is the production of stuff higher up the food web. You don't care how much algae is sitting around. You care how fast is the algae producing food that the zooplankton can eat, and how fast are the zooplankton producing more zooplankton biomass that is food for the planktivores and so on. So that's a different type of ecology question. And we look at both of those, and a big question that I think is really interesting is what is the annual cycle of algae? I mean, we're getting ready for spring right now, and you look outside and, okay, there's snow today. It looks like winter again, but the snow's going to melt, right? And the birds are singing and buds are starting to swell up on the elm trees and the maples, the early flowering trees. We know what the seasons are up here on dry land. Birds singing in the springtime and flowers coming out, leaves bursting, and, you know, baby deer getting born in the woods and baby rabbits, and, you know, spring and summer, and we know fall falls in the, we know winter, but what are the seasons for a lake? Do lakes have seasons? I mean, under the ice, the water is always liquid. You know, it's not like up here on dry land where the water is liquid in the summer and it's frozen in the winter. You know, and like if you're a tree and you're trying to get water out of the ground, it's not going to happen in the winter because the ground is frozen. Really big difference between summer and winter if you're up here on dry land. But under the ice, it's always about freezing. There's always liquid water. If there's no snow on the ice, there's sunlight. If there's nutrients in the water, they're still circulating around. So it's not really completely clear what would the seasons for a lake be. They're going to be different from our seasons up here on dry land, but will there be seasons? Is there a predictable pattern? Is it repeatable? Do eutrophic lakes have one kind of annual cycle and oligotrophic lakes have a different cycle? Do lakes in the Arctic have one kind of cycle and, you know, seasons? Do lakes in the tropics have different seasons, you know? So that's a big question that we're going to look at. And when you think about the seasons of a lake, you really are thinking about the phytoplankton. But you're also, I mean, thinking about the zooplankton. I mean, the zooplankton eat the phytoplankton, so the phytoplankton affect the zooplankton. But also the zooplankton eat phytoplankton. The zooplankton affect the phytoplankton. And of course, the phytoplankton are eaten by fish. Little fish are eaten by big fish, which are eaten by bigger fish. And, you know, fish, many types of fish only reproduce once a year. Most fish lay their eggs in the springtime. And so in the springtime, there's going to be lots of like tiny little larval fish. And then in early summer, little juvenile fish. And then by the end of summer, maybe bigger fish. So the fish have an annual cycle up here in the food web, and that's probably going to have an influence downstream too. So the seasons of a lake, the annual cycle in a lake is not random, but interesting. It's got different levels connected to each other. So I think that's what we will lead up to today. So I think I got these kind of three big topics that I'm gonna talk about today. Not necessarily in that order, but we're gonna touch on all of them. Another thing we're gonna talk about today is looking ahead, what we're gonna do, because I know that some of you have been practicing your rope tricks. Here's a great big rope tied in two hanks and wound around in the middle. It's kind of hard to tell because this rope is multicolored. But here's some old telephone wire I had in the basement, and notice how beautifully it was wound in a coil, and then with a wrapping around the middle, just like you would do with a rope, except it doesn't unspool very easily, so it only needs just one or two wraps around the middle. And looking at my schedule here, it seems to me that we have a week of lecture, and then next week we have our second exam. Yeah, completely out of sync with my other classes. So we got to talk about that too. Looking ahead, planning lab for this week, and talking about what's going to happen next week. All right. I think what we want to start with is planning Thursday of this week. All right. 2 weeks ago, I was extremely optimistic, very optimistic, that we would be able to get one more lab on the ice. And then the weather forecast got worse. Yeah, the weather forecast changed. The weather forecast became warmer, sunnier, less cold. And the forecast for the ice, therefore, worsened from our perspective. According to my ice prediction model, ice condition prediction model, forecasting model, the ice on Thursday of this week will be unsafe. Now that might not be true. The ice might in fact be safe. But when it gets to the point where it might be unsafe, I have to say I'm going to assume that we will not be able to go on the ice. Yeah, I'm really disappointed by that, because I had grand plans for what we were going to do. We're not going to do that. We're going to do something simpler. What we're going to do is, as scheduled, we're going to have the Lab 6 lab practical and a little bit of experiential observation of rotten ice. If you have never seen rotten ice, or never seen a moat around a lake, hopefully we will, at this point now, I'm hoping we will have both a moat and rotten ice, honeycombed, as they call it. And for those of you who have never seen that, this will be a valuable experiential experience. And for those of you who have seen it, well, it's beautiful. So what we're going to do on Thursday of this week is we are going to meet at our usual place at Lake Phelan Boat Ramp. But we're going to divide into 3 groups. We have about 18 students, and we have about 6 sets of equipment. So when I'm going to do subsections, whatever you want to call it, 3 groups of 5 or 6 people each. The first group will come to lab at 1 o'clock for 1 hour. The second group will come to lab at 2 o'clock for 1 hour. And the third group will come to lab at 3 o'clock. Oh, that was simple. 3 o'clock for 1 hour. How are we going to divide into groups? Well, some people may find it easier to come to class early, and some may prefer late. So what I did was I created a survey in D2L. Well, let's go to D2L, and I'll make sure that it's still there. Jeffrey's going to tell me if this works. Yeah, here's how the D2L page looks to me. Here's my ice forecast. Updated this yesterday. The model predicts rotten on the 18th, which is Thursday, and ice out the first windy day after that, or the 21st, whichever comes first. I think Saturday and Sunday are going to be windy, so I'm predicting, my model is forecasting that the ice will leave the lake this weekend. I'm checking my calendar over there. Yeah, Saturday or Sunday, probably Sunday, just guessing. Okay, so go to the D2L page, go down to surveys. Yeah, here's a survey. And I got a survey down. Oh, I better make this visible to you. Here we go, make visible to users. Here's a survey. It's called Lab Section Preference for Thursday, 18 March. So go to the lab survey right now and tell me, what's your first choice for your 1 hour time in lab on Thursday? Do you want to come to lab from 1 to 2? Do you want to come to lab from 2 to 3? Or do you want to come to lab from 3 to 4? 1 hour each, starting at 1, starting at 2, or starting at 3. What's your first choice? I'll try to give everybody their first choice if possible. What's your second choice? I'll try to give everybody their second choice if they can't have their first choice. What's your third choice? And that would be the one you don't want or you're least attracted to. And then there's a meh button. So like if it's all the same to you, just say you have no preference and that'll make it easy. So please go out to the survey right now and essentially rank those 3 times for when you will come to class on Thursday, 1 o'clock, 2 o'clock, or 3 o'clock. So go out there and do it, and I'll give you, a couple minutes to think about this and fill out that survey. Any questions about this?
Can you go over what's going to be on the lab practical?
Yeah, what's going to be on the lab practical? Let me pull up the calculator. Let's go back to Zoom, share my screen. Okay, this is posted on D2L in the lab manual. This is lab 6, Limnology field skills. This is what you have, this is what we're talking about. Learning to use the GPS, compass, rope, and surveyor's line. Here are your learning goals. These are the things you need to know about the GPS and UTM coordinates. Remember, we did a whole lab where you used the GPS and 2 different labs in which you used UTM coordinates. Compass direction and triangulation. We did that in our last lab, the triangulating using the compass. We used a surveyor's line in the very first lab, and we went over last time. I demonstrated in lecture last week, or whatever, the week before this one, to tie lots, and then we practiced them standing around the boat launch and last lab. These are the learning, these are the skills that I hope you have mastered. So a list of them, and this will be evaluated with a practical test. So I could, for example, I could give you a rope. I will give you a rope and say tie a square knot, or give you a rope and say tie a bowline, or give you a rope and say coil this neatly and secure with an appropriate knot. I could give you a compass and say what is the direction from here to that landmark over there, in letters and in degrees. Okay. So I think this says exactly what you need to be able to do on the field test. Obviously, you won't test all of them because there's like 20 things here, 15, 10. Oh, I guess I could, there's only 10, 5, 10, 15, okay, whatever. Does that answer the question?
Yes, thank you.
For me, I got a lot of stuff in the lab manual, so I got to like scroll down and say show more, but it's in there. All right, I'm gonna just slip out to D2L and see what is it gonna tell me. Can you tell me how many people who have responded here?
There was a question on the chat. Were you?
Oh, okay. All right, so let's see, 15 people have answered the survey. So that's probably all of you. Yeah, no, I don't know exactly how many I will test you on, but it could be any, it will be more than one. Presumably at least one from each category. Yeah. And this should be nice. I know that some of you are really tired of you know, answering questions on pieces of paper and writing up lab reports. So this is instead of a lab report, this is how you get credit for lab 6. So that should be a nice change for some of you will like the change, I think. I'm confident of it. All right, so it looks like, looking at the survey, we've got 6 people prefer 1 o'clock, 9 people prefer 2 o'clock. Wait, is that right? It's not really quite right. I got 6, 3, and 3, and 3 people are fine with anything. So that's really close to 5, 5, 5. So this will work. And hopefully by the end of, maybe even by the end of class today, I will be able to tell you what, or certainly by the end of today, I'll tell you what section you're in and therefore what time to show up for lab on Thursday. Lab on Thursday will take 1 hour. You will either come at 1, 2, or 3 o'clock. And the main activity will be the lab 6 lab practical. And the other activity will be a little bit of looking at the lake and the ice and talking about the ice condition, which is information you want to have in your head 2 weeks from now. 2 weeks from now when we talk about lake ice and heat. Okay. Lab this week, looking ahead. Hey, while we're looking ahead, next week is the midterm, the second midterm exam. And on Tuesday, we will be reviewing and discussing a research article The research article for next week is posted on D2L in the readings section. And the article is by Professor Shapiro, Joe Shapiro, and Dave Wright. And it's Dr. Shapiro was my limnology instructor and one of my professors at the U of M. And this is research that he did with a grad student, Dave Wright, on a lake in the Twin Cities metropolitan area. So that article by Shapiro and Wright, it's fairly short, is posted on D2L, and that is your reading assignment for next Tuesday. We will use
you
that.
have a study guide for exam 2?
Possibly. The study guide for exam 2 will look just like the study guide for exam 1. Oh, so it should be pretty easy for me to write. It's going to look just like the study guide for exam 1.
But adding 5 more weeks.
Adding 5 more weeks. Because we've gone over and over again the stuff that we learned in the first unit. Yeah, the first 5 weeks. And then you, of course, you have reading questions for each chapter. So you have a very detailed study guide, pretty detailed study guide already. Okay. Well, that's pretty good. Any other questions about next week? So Tuesday we'll review and we'll discuss the article by Shapiro and Wright. I will put a Q&A survey up. So if you have any review questions that you want to get answered during the review session, put them in the survey. I'm pretty sure we did that for, did we do that? Yeah, I did that for the exam 1. We'll do the same thing for exam 2. Just if you have any questions that you want to be sure get answered during a review session, put them there. All right, well, that's a lot of talk. Any other questions? Go back to Zoom so I can see your smiling faces. Smiling names. Well, you know, some of you are smiling in your pictures. All right. All right, well, let's have a little lecture then. So what we're going to talk about is algae. And we're going to start with a blue slide. Yeah. Algae part 2, phytoplankton ecology. So you'll recall that last week, the last week you had class, we talked about different ways that algae are categorized. Algae can be categorized kind of taxonomically, you know, which is about the family tree, you know, what's related to what. You know, the cyanobacteria are all closely related to each other. Diatoms are actually closely related to the chrysophytes. You know, they had a common ancestor not that long ago. Cryptophytes and euglenas are small unicellular things, but they're really, really different from each other in terms of like their ancestry. So we can talk about the different types of algae by their taxonomic group, which is the way I think a biologist maybe would look at them, like a cell biologist or organismal biologist maybe. But another really sort of different but important way to look at the algae is to look at them in size classes. Are they net size or are they too small to get caught in a net? That's just a very practical thing about the algae. Like, how big are they? Are they so small that they slip through the net of a zooplankton net and you never even see them? Or are they big enough that they get caught in a net? Or more fine tuning it, are they teeny tiny little algae so small you can barely see them with a microscope? Or are they great big algae like kelp, that make forests? Or are they just the perfect size for Daphnia to eat them? Or are they like too big for Daphnia to eat? Are they so small that rotifers eat them? So the size of the algae alone, regardless of their taxonomy, is an important way to categorize the algae. And it has important consequences for the place that the algae fit in a food web, their interactions with other species, and therefore the types of lakes in which you find them or the times of year in which you find them. So you can classify the algae, the phytoplankton, by taxonomy, and there's an incredible diversity, taxonomically speaking. Everything from prokaryotic cyanobacteria to obscure things like cryptophytes, to things like green algae, which are closely related to plants. Right? So lots of taxonomic diversity. We can categorize them by size. And in some situations, size is just simply the most important thing. Or we can categorize them by how they fit in a food web, how they interact with an ecosystem, how they function in a sense. And this is often called the functional groups of algae. You could call these guilds, if you prefer the word guild, but lake ecologists don't usually use that word. They call these functional groups. And those functional groups, they might be 6 or 7 or 5, depending on how you group them, but we're gonna say that there's 6, unless I'm counting wrong again. Yeah, so what do we got? We got the blue-green algae. Some can fix nitrogen and some can't. We got the diatoms. They're fantastic and they're unique and they're important. The silica cell walls. The big green algae, things like volvox and spirogyra and Edygonium, big filamentous mermaid's hair green algae. Dinoflagellates, which I've mentioned before as having some weird properties. And the pigmented flagellates, which is kind of like everything, small, unicellular with a flagella. And I guess it's 6 if you divide the blue-green algae into nitrogen fixers and not. Okay, so these are the functional groups of algae. And I've said a few words about each of them. But we really want to review these because we're going to talk about these and how they fit into the ecology of things. So what I've done here is I'm going to create 5 discussion groups. And I want each group to be responsible for one of these groups of algae. Breakout room 1 is responsible for the blue-green algae. Breakout room 2 is responsible for the diatoms. Breakout room 3 is responsible for the colonial green algae. Breakout room 4 is responsible for the dinoflagellates. And breakout room 5 is responsible for the pigmented flagellates. So Zoom is just going to randomly assign you to a breakout room. Someone in your group, you need to appoint a chair, and the chair is the person who knows what the assignment is. And here's the assignment. For your group of algae, we want to know the name of the group, and this would be both scientific and common. The scientific taxonomic names. We want to know the key characteristics. Does it have cellulose in the cell wall? Does it have silica in the cell wall? Does it have flagella or not? Is it prokaryotic? Is it eukaryotic? Does it have like a oopy protective gelatinous sheath around it filled with chemicals? Does it make famous toxic chemicals or not? Right? You know, what are the key characteristics? Some of them have a particular shape, some don't. We want to know one or two famous species or genus. And I want a picture, which is to say a cartoon. A cartoon of it, you know, like a little diagram. You know, next week when we read Shapiro and Wright, several species of algae are referred to by name. And they're famous genera that any limnologist would know. So you need to know the names of some genera. And for your group, you want to tell us what the most famous one or two or three are. These are going to be ones that are in the reading questions as taxa you need to know. These would be ones you see in the diagrams in CALF. These would be the ones that you see mentioned in the paper by Shapiro and Wright. I kind of leave it up to you. So maybe there's a little guesswork, but make sure that at least belongs to your group. And then we want to know, you know, does it have any important ability? So not just characteristics like physics, but characteristics I kind of mean like physical structures that you would show in a diagram, like a flagella or not, right? But then there are also things that you can't really show in a diagram, but are important abilities, like can it swim? Can it swim? Can it swim phototactically in response to light? Can it control its buoyancy so that it can float up to just the right level or float down? Can it fix nitrogen? What would other important abilities be? Can it also live heterotrophically, absorbing organic material from the water? Right, so both phototrophic. Is it ever mixotrophic? So those would be like important abilities. Okay, so that is your assignment. Every group needs to appoint a chair. The chair is going to be the person who can look up at the top of the screen and see what number you are in, what your breakout room is, and therefore know which group you're responsible for, and know this is what you're going to do. So you're going to create a whiteboard or a shareable document of some kind that you can show us. We want this information on it. There's going to be a picture, some kind of cartoon, and I want the whole thing bullet points. Don't write an essay. We're just looking for, 20 words. So this is your assignment, 20 words, 50-50 mix of words and pictures. Which you may recognize as part of the 10-20-30 rule and the 50-50 rule. And since you're only going to be giving a 1-minute presentation, you get one slide, right? That's all you're talking about. Okay, do you have any questions about your assignment? All right, I'm going to let's see how this goes here. Okay. Excellent. We have 5 rooms created. Every room has 3 people in it. Every room needs to appoint a chair. So hopefully at least one of the 3 of you knows what this assignment is. If you're in room 1, you're doing blue-green algae. If you're in room 2, you're doing diatoms. If you're in room 3, colonial green algae. If you're in room 4, the dinoflagellates. If you're in room 5, pigmented flagellates. So appoint a chair and then a note taker, somebody who can create this visual aid that you will then share with the class. Okay, so the purpose of this is to review the 5 functional groups of algae and the taxonomic groups within it and their important characteristics in preparation for talking about the ecology of algae. Okay, the rooms are open. It's kind of a big question. I think this is a 10-minute question. And while you're doing this, I'm gonna get Thursday groups figured out.
So it looks like we got dinoflagellates.
Okay, yeah.
Yep.
Does anyone want to be the chair?
Mm,
I don't really, it doesn't matter to me, but I cannot do a whiteboard right now on my computer.
Are you okay if I just screen shared? Would that be acceptable?
Yeah,
for
yeah,
you
that's
guys?
totally fine.
yeah.
Okay. All right. Do you guys see this?
Yeah.
Okay.
Yep.
And then I wrote down what she told us that she wants us to look for. So that's right there.
Okay.
I'm just gonna grab my textbook real quick.
Sure.
So has she already given us a scientific and common name for the group?
I think just Pyrophyta.
Yeah. So for the key characteristics, there's like the 2 flagella.
Okay.
Then for important abilities, I think the cellulose plate armor, would that be an ability or is that like a characteristic?
What was it again?
The cellulose plate armor?
I think that would be a key characteristic,
Oh, okay then.
just because that's like that's more of a part of its like body, like its physical structure.
Okay, yeah, you're right.
Could you say that one more time for me though? Sorry.
It's okay. Cellulose plate armor.
Okay. Pre-armor?
Plate.
Plate. Okay.
Yep.
Yeah.
Then its abilities is that it could bioluminesce in oceans.
That's pretty cool.
What page are you reading this from?
It's from her lecture
Oh, okay.
presentation thing. Yeah.
Do dinoflagellates have a certain shape?
It looks kind of like a spaceship.
Oh, it does. Look at that. Okay. They have a split. I guess they have a spinning flagella. Oh, this is the one that creates red tides. Cool. The scary one. Would spinning flagella also be a key characteristic, or would that be an important ability?
It'd be both. It's motile for the ability, and then the characteristic would be the flagella.
Okay.
It's unicellular. Do we have that?
There we go. For the one or two famous species slash genus, what would that be?
It looks like peridinium and keratinium.
Oh, okay. Right there.
I think those are the two genuses.
I'm gonna have to figure out how to spell that. I'm just gonna take a screen snip of it.
Sounds good.
Okay.
They're asexual. It looks like they reproduce by division.
Okay.
Apparently, the one that's in the lecture, the picture of it is the Ceratium one.
Okay, cool.
Okay. Do you guys
Cert.
want me to try to do like a picture of a Ceratium then?
Yeah.
Okay. Hmm.
In the Calf book, it also says that it's heterotrophic.
Heterotrophic?
Yeah.
I'm trying to find something about the size right now.
What page are you on in the Calf book?
I'm looking at 327 right now.
Okay.
That's where the size stuff is, but I'm finding like multiple types or whatever. So it looks like they're larger. I think they are net caught or whatever.
Or they're net
Oh wait, never mind. Now it's saying nano.
nano? Okay.
Yeah.
Would it be, let's say nanometers?
I think just nanoplankton. They just have like 3 categories of sizes. But yeah, nanometers.
Great. Any last minute ads?
I think we're good.
Okay. Sounds good. I'll stop screen sharing.
All right, welcome back. I'm smiling because Jeffrey, you know, he doesn't hear anything, he doesn't say anything, but he's visual, and he thought his group was doing a great job. Looked like they were really constructing something good. So I'm looking forward to this. All right. Well, let's start with group one. Group one had the biggest, I think, bunch of things, which is to say the blue-green algae. So Ruhwad, Abdul, Christina, Sam Thomas, tell us about it.
See, so we had blue-green algae, which is also a common name. Scientific name is cyanobacteria. A few characteristics that we listed were prokaryotic. There is no flagella, come in gelatinous sheaths, and they are sometimes toxic. A picture, we didn't know how to put a picture into the whiteboard, but we drew them. Really, it's just kind of some small little green dots for one of the species is Microcystis aeruginosa,
Sure.
something like that. Abilities that we listed were controlled buoyancy with gas vacuoles, and then obviously, as we listed before this little assignment, blue-green comes in nitrogen fixing and non-nitrogen fixing. So the species that we listed as nitrogen fixing would be Anabena and then Aphanozomenone, something like that. And then non-nitrogen fixing, as it's the one that we listed the picture of and drew, which was the Microcystis.
Yeah, well, that's great. That's a nice picture you drew. And you know, you struggled with the names that are hard to pronounce, so no shame there. Anabena, Aphanizomenon.
Anazomenon. Oh, there we go.
Aphanizomenon. Although I have to admit, some people call it, I think it's a joke, but they call it aphanizomenon, which is like backwards with every syllable almost. And you spelled them all correct, great, and that's a nice diagram. And those are the key characteristics of blue-greens. Anybody have any questions about the cyanobacteria? All right, nice job. Thank you very much. Nice job, really. All right, let's go to group two. Room two was Bree, Zhengli, and Ju. And you guys had the diatoms.
Oh, okay, yep. So we got diatoms, the scientific name for it. I don't know how to pronounce this. Baxillerophyta or something. And then some of the key characteristics that we listed were that they create very unique geometric cell shapes, which derives from like their cell walls. They have graph, which allows them to move. They're phototrophic, mesotrophic. They have a distinct cell division, and they don't have flagella. They're unicellular, and they could form chains, colonial chains. We also forgot to mention that they're eukaryotic. And then the example we showed here was Aceronella. We got this picture off of Google. And then some important abilities that diatoms have are that they have a cell wall that's made out of silica. And there's like two valves that gets pushed outwards when there's cell division. But then what's unique about the cell wall is that the silica makes it heavy, and so the diatoms are able to sink faster throughout the water column versus other phytoplankton. And then some diatoms can fix nitrogen, but that's through the assistance of an endosymbiotic relationship with other organisms. And then they also store energy as chrysolaminarian. I think they can do it with cellulose too, but that would be a waste of energy for them.
Yeah, all right, that's great. And thank you very much. Nice job. Nice job. Yeah, so the diatoms, Bacillariophyta or Bacillariophyta, and mostly they're not mixotrophic, but I think some are thought to be. And mostly they're not nitrogen fixing, but there are some that have an endosymbiotic cyanobacteria that fix it. But otherwise, I think you got the key features. Were there any questions about the diatoms? All right, let's go to room 3, which is Brett, Desiree, and James, who had the dinoflagellates.
Can you guys see that? Can you guys hear me? And the key characteristics are that there are many types. Some are unicellular with flagella, and some are unicellular without. Some form a plate colony, and some form spherical colonies, and some have filamentous branching, and they all have a cellulose cell wall. Famous genuses that we found were Volvox and Cara, and I have a picture. This is the Volvox. And some important abilities were phototrophic, and they can reproduce by cell division or sexual reproduction, and they store starch for energy, and they can actually be different colors, like not just green. They have a range.
All right. All right, nice introduction to the green algae. It sounds like you had a picture, but in a different file.
Yeah, can you not see this?
Yeah, what you have to do is you have to stop sharing this and start sharing the new one.
Okay.
Yeah. I just learned that too.
Here it is.
Yeah, it's a Zoom thing.
Yeah.
And there you go. And there's a nice full box. All right, thank you very much. Nice introduction to the green algae. Any questions about the green algae?
Yeah, Brianna.
I think she's just applauding.
Oh.
Yeah.
I'm just applauding. I'm trying to be supportive.
Yeah. Good job, people. Yeah, like I said, public speaking, if it's like on Zoom, it's like, I don't think so. All right, let's go to the next room. I think I got it. I think I have the list in a different order than I had on the PowerPoint. So let's go to room 4, Hannah, Kat, and Tiffany. And you guys had, and you guys had that.
Yeah, we had the dinoflagellates. So yeah, the name for it was the Pyrophyta, or the dinoflagellates. Key characteristics are they've got 2 flagella, they've got cellulose plate armor, they've got a spinning flagella, they're unicellular, and they reproduce through division, so they're asexual and they're heterotrophic. Important abilities are they're bioluminescent in bodies of water, so these are the ones with the red tides. They're small, nanoplankton. And then the species that was attempted to be drawn, but then we also have the picture next to it is the Ceratium, the Caratium. But yeah.
All right, that's a nice introduction to the dinoflagellates, which they're a unique, and everybody always agrees as to who is in the group, but there's a lot of different names for that group. Some people call them Pyrophyta, which I think is related to the Greek, Pyrrhic victory, meaning setting things on fire, referring to the neurotoxin. capability of it, but other people call it the dinoflagellates or the dinoflagellophida or the dinophyta, some version of that, which is really referring to their spinning behavior as they swim. So it's a well-defined group, but the name of the group, I think even in CALF, even in the textbook, you see it referred to by 2 or 3 different names, depending on, you know, the diagram or who CALF is citing. Any questions about the dinoflagellates?
I have a question. I was in the group, but are they found in large and small sizes? The CLAF textbook was a little confusing about that.
Yeah, the one that you're looking at right there, the serratium, as people say, or probably technically should be keratium, but people say serratium, is actually pretty big. So they do have kind of a range of sizes. They're always unicellular, but some of them like this are actually quite big because of the cellulose spikes.
Okay, thanks.
Yep. And the famous ones have these like great crazy cellulose shapes, but there are some others that are just little unicellular looking things, you know, not very distinctive looking. The ones that are symbiotic within coral, for example, are just don't have the cellulose armor, not very distinctive looking. All right, let's go to room 5. Speaking of small, not very distinct looking things, and Dolly Keenan or Sam?
All right, let's see, I made a small PowerPoint. I thought that would just be the easiest way.
You know, whatever works for you.
Can you see that?
Yeah.
All right. So we have the pigmented flagellates, which contains the Cryptophyta, Euglenophyta, Chrysophyta, and then the unicellular Chlorophyta. Characteristics are that they are eukaryotic. They all possess flagella. They have photosynthetic pigmentation, and they are typically mixotrophs. For important genera, really the only one that I knew, like new was Euglena. They're the one that I was the most familiar with, and I figured that the most people would be the most familiar with. And some abilities are they can swim via their flagella. They often have light detection, like Euglena has the tiny little red eye spot there you can see by the flagella. And with those 2 abilities, they can exhibit phototaxic movement. They move towards where light is so they can feed more easily. So.
Excellent, excellent. Very nice job. Thank you very much.
Yep.
Nice job. I like the PowerPoint too. I mean, that's a good idea. I mean, you can draw in it, you can copy and paste in it and stuff. I mean, why not? Okay, so we have reviewed the 5 or 6 functional groups. If you separate the cyanobacteria into nitrogen fixers and non, then you get 6 groups. And often that is a reasonable thing to do because that nitrogen fixing ability is really, really special and in the key ability in some circumstances. All right, so now let me show you a little PowerPoint here somewhere. Go back to my PowerPoint and start talking about the ecology of these groups. Let's see. Get everything queued up here. Okay, so we got 5 or 6 big functional groups, and you know, whatever 7 or so taxonomic groups. Is there any pattern to where you find them in the world? Is there any pattern to which lakes? Like if you wanted to find cyanobacteria, where would you go? If you wanted to find chrysophytes, where would you look? Well, one big thing that causes that is strongly correlated to this question is total phosphorus. So what we have here is on the x-axis, total phosphorus. This is micrograms per liter, which translates to parts per billion. And you can see the difference between a very oligotrophic lake is, you know, down there in the single digits, total phosphorus, and a eutrophic lake, double digits. That's the difference. The difference between an oligotrophic lake and a eutrophic lake is the difference between 5 and 50 parts per billion, micrograms per liter. Anything over 100 and it's like seriously eutrophic, like up there in, you know, Como Lake territory, like dystrophic, like yuck, green, oh, pea soup. And what do we see? In oligotrophic lakes, there are a variety of different types of algae that you will find. You get to very eutrophic lakes, like anything over about 100, and you know, pretty much 100% of the biomass is going to be cyanobacteria, blue-green algae. And as the phosphorus rises, so does the tendency for cyanobacteria to dominate. Some, on the other hand, species like the chrysophytes and the cryptophytes, you tend to find only in very low nutrient water, clear, low nutrient, not a lot of other algae there. That's where you're going to tend to find the cryptophytes and the chrysophytes. Now, this may be one reason that we don't see a lot of them in limnology class, because we study lakes right here in the Twin Cities, which tend to be, well, they were mesotrophic to start with, and then a city got built around them, which is going to make them eutrophic, just because people use fertilizer on their lawns, you know, and some of it washes into the lake, and leaves fall, and they, you know, they get driven over and broken up, and they wash in with the stormwater, bringing nutrients with them. So we don't see a lot of chrysophytes or cryptophytes in class because we don't study oligotrophic lakes here in lab. We see a lot of cyanobacteria. We see a lot of diatoms. There are different diatoms at every sort of, almost any kind of range of trophic status, as they say. So this is a key diagram. I hope that you read it carefully. You'll notice this is Lake Erkin, Sweden, and there's other information about Lake Erkin scattered throughout the textbook. It's a well-studied lake, and there's been a lot of good ecological study of Lake Erkin. So I think we've seen data from Lake Erkin before, and we'll see different stuff from it in the coming weeks. Here's another way of looking at this business of trophic status and blue-green algae dominant. This is an interesting diagram, the way it's constructed. You'll notice that the x-axis is total phytoplankton biomass. That correlates strongly to total phosphorus. It correlates strongly to trophic status. In fact, it's almost the definition of trophic status. Oligotrophic lakes have a very low total phytoplankton biomass. Not a lot of phytoplankton, clear water. Eutrophic lakes, on the other hand, have a lot of phytoplankton. So the x-axis here is labeled total phytoplankton biomass, because that's what they measured. But this x-axis is actually very similar to this x-axis. Oligotrophic on the left, eutrophic on the right. Oligotrophic on the left, eutrophic on the right. Okay, so on the horizontal axis, we have the total phytoplankton biomass, and this is like milligrams per liter. On the vertical axis, we have a very, very similar variable. Biomass measured in milligrams per liter, but not total biomass, just the biomass of the blue-green algae. Now, if blue-green algae were 100% of the algae in all lakes, then this line here, the line that is in red, is the only thing you would see. This line here in red is the one-to-one, the one-to-one line. See, look, on the left-hand side here, where it meets the y-axis, here we have 0.01 milligrams per liter, and the vertical coordinate of that point here is 0.01 milligrams per liter. So if 100% of the algae was blue-green algae, then your data point would be right there on this line. Now look at the other end of the line. Here at this other end of the line, this data point here where the arrowhead is, that point here would be a lake or a water sample in which there was 10,000 milligrams per liter of phytoplankton and 10,000 milligrams per liter of blue-green algae. So this top line, this diagonal line here, is the 100% blue-green algae line. And since you can't have more than 100%, there's nothing above the line, right? If you have 1 milligram per liter of phytoplankton and all of it is blue-green algae, then your data point's going to be right here at the 1. Okay, so I guess I feel like this is a hard diagram to read, which is why I've taken the time to put it into words out loud and stuff and describe it. You want to just like give me a thumbs up if you want, or give me a check mark if you feel like you understand this diagram. Give me like an X if you still don't understand, and I'll just see how you guys are doing. Check mark if you feel like you can read this diagram now. X if you're like no. I got a bunch of checks and a couple of X's. Okay. Well, I'm going to say take some time to look this diagram over, and I'll tell you a little more about it now. Okay, this diagonal line is the one-to-one line. You know, the y equals x. That's what this line is. And in every one of these lakes that's on this line, all of the algae was blue-green algae. Here was a lake that had a huge amount of algae, and 100% of it was blue-green. Here's a lake that had really not that much algae, and at that time anyways, all of it was blue-green algae. And what you can see in the overall pattern of this is that when there is a lot of algae in the water, a lot of phytoplankton in the water, all of the water samples are up there on the one-to-one line. In the water samples in which there was more than 100 milligrams per liter phytoplankton, all of that phytoplankton was blue-green algae. When you get down to less eutrophic lakes, lakes with less algae, less phytoplankton in the water, there's a much greater range of how much of the algae is blue-green algae. Ranging from all the way down here, this is the 1% line. Down here, less than 1% of the algae was blue-green algae. These samples in here between 1 and 10%. These samples in here between 10 and 100. As you get to lower amounts of phytoplankton in the water, it's less sure. It's less of a sure thing that the cyanobacteria will dominate. There could be a lot of cyanobacteria, there could be very little, you know, quite a range. But when you get to eutrophic lakes, they're all 100% of it is blue-green algae. Now, this is figure 21.6. This is in the textbook. You can read a little bit more about this at your leisure. This set of lakes is not a Scandinavian lake, Lake Gherkin up in Sweden. This is a set of lakes in Florida. So it's a very different group of lakes, but we see the same pattern. we see that when it's very eutrophic, blue-green algae dominate. When it's not very eutrophic, they might or might not. And so in a sense, that's a similar pattern to this. When you get up to the high concentrations, the very eutrophic lakes, yeah, the blue-green algae will dominate. So these two diagrams kind of show you the same thing. And here's that microcystis again. Yeah. Well, let's look at something at the other end of the extreme. That would be like chrysophytes and cryptophytes. And here's cartoons of two chrysophytes. And I think perhaps you remember me saying that when there is a low, in an oligotrophic lake, that would be a low biomass of phytoplankton. That's the only time when you really see substantial amounts of chrysophytes. You get up even to mesotrophic lakes, and the chrysophyte biomass pretty much just drops to zero. It's just really, you just don't see. Now, the exception is probably this one, Dinobrian or Dinobrian, if you prefer. This one here, we do see this in class sometimes. And so I think it's maybe the most mesotrophic of the chrysophytes. It's kind of almost an exception to the chrysophyte rule. But the general rule is oligotrophic lakes towards the left, mesotrophic in the middle, eutrophic heading off to the right. You're only going to find that chrysophytes are a significant percent of the algae in the very oligotrophic lakes. And this is percent of the algae. This is not even total biomass of chrysophytes. This is what percent of the phytoplankton are from this group. And it's only ever more than 10%. Here's the 10% line. It's only ever more than 10% when you're down below on this chart, like 5 milligrams per liter, right? 5 milligrams per liter of phytoplankton on this diagram would be like way down here, just for point of comparison. And that is kind of like zooming in. Here are the chrysophytes here. And here, what percent of the biomass comes from that group. And you're only going to see, what is this, 10, 20, 30. This is just a summary cartoon. But here they're showing 30% chrysophytes down near total phosphorus about 4. It's very low. Okay. So that's kind of the two extremes, chrysophytes and cryptophytes, only when it's very oligotrophic. When it's very eutrophic, it's definitely going to be cyanobacteria. Abdul.
So is there a reason why there's so much more chrysophytes at that low phosphorus content? Are they like being outcompeted by cyanobacteria, or is like phosphorus harmful to them?
Phosphorus is not harmful to them. Why are they abundant only in very oligotrophic water? You know, I don't actually know why that is. I know reasons why cyanobacteria dominate in high phosphorus waters. But I think that when you get to low phosphorus waters, right, you see quite a diversity of different things. And it may be, I don't know how to put that into simple words, but I think that's kind of the big picture here is, in fact, in low nutrient conditions, you get a diversity of algae. When you get to very high nutrient conditions, high productivity, the diversity drops out and you end up being dominated by a very, very small number, and even a small number of species within the cyanobacteria. It'll be different in different lakes, but the diversity totally drops off when you add nutrients to a lake. And that might be the opposite of what you would expect. Like some people say the rainforest is really diverse because it's whatever high productivity, and that's just not true. It's not a generally true statement that higher productivity leads to diversity. In fact, very high productivity often leads to a loss of diversity. Like if you want to get rid of the weeds in your lawn and just have your lawn, if you're a homeowner or a golf course manager, if you want to have your lawn be nothing but like Poa pratensis, like one species of grass. Yeah, you can put down weed killer, but even better than that is fertilizer. If you super fertilize your lawn, you end up with a monoculture of one species. Just like you see, well, not exactly, but a lot like what you see in lakes. You fertilize them and you end up with just one species. So adding nutrients, increasing productivity is not as a universal law, as some people think it's like a law of nature. It's totally not, because it's not true at all in lakes. Anyways, I'm digressing because I believe I'm at the end of these 3 slides of this topic. And the next topic is biomass versus production. So this is a different topic. So I'm going to stop sharing because it's time for our break. I took the survey results that you gave and I constructed 3 groups. These are all people who said 1 p.m. was their first choice. Abdul, James, Kat, and Dolly, Bree, and Jane. The 2 o'clock group, Hannah, Desiree, Christina. Brett, I think, don't you usually get a ride with Christina? Hannah, sorry, so I moved you to Hannah's time slot.
Yeah,
you
that's
said
perfect. Thank you.
Because you didn't get a chance to talk to each other. And Tiffany, you said you didn't care, so I put you here. And then 3 o'clock, Keenan, Ju, Sam, and Saul is absent today, so I just put him in that group. So these are the assignments for Thursday. If you realize you made a total mistake and you can't go to that time, there's a time slot open here and here, so you could switch and tell me that, whatever, by the end of today. Like if you realize it after class, you're like, oh my God, on Thursday I have a dentist appointment at 1 o'clock, whatever. Email me after class today and I will post it either tonight or tomorrow morning. Any questions about, any questions about Thursday? Everybody good? These work for you? Give me a yes if this works for you. We'll just get a quick look. If you're like, oh my God, no, you can give me a thumbs up if you're really happy. Because you're like, oh yeah, only 1 hour. Oh, I'll be free at 2 o'clock. I can, whatever, get lunch. My life will be so improved. Okay, I got no frowny faces and no X's, so this all looks good. But again, think about it during the day today. If you've realized you made a horrible mistake, email me tonight. Okay, 2:30, time for our coffee break. We're going to come back and talk about more phytoplankton ecology topics. So, 10-minute break. Oh, what? Oops. La la la. Okay. Oh, what did I do? Well, now. Okay, that's very interesting. Yeah, okay. Here we go. Jeffrey's going to show me if I got it. There we go. Okay. So we talked about the fact that lakes that are very eutrophic, you really just get dominated by blue-greens. It's just almost always true. And that you really, if you want to find chrysophytes, you really need to go to oligotrophic lakes and cryptophytes too. We could have, if we had organized today's agenda differently, spent a little bit of time on this question here. I don't want to spend a lot of time on this, but I want to give you a chance to connect what you reviewed with this topic. So I'm going to put everybody back in their breakout rooms for just 3 minutes, 3 to 5 minutes, and to see if you can figure out the answer to this question. Blue-green algae are often the most abundant type of algae in eutrophic and hypereutrophic lakes. Like, it's an extremely reliable thing. Why? So what special traits do blue-green algae have? And which of those help them dominate in eutrophic and hypereutrophic lakes? And I will tell you that there's more than one reason. There's more than one special trait that blue-green algae have, but there's maybe 3, or at least 2, maybe 3, okay? So everyone's gonna have the same question. Why do blue-green algae dominate in eutrophic and hypereutrophic lakes? Did you let those breakout rooms that you had before look pretty productive? Do you want to keep them? Give me a yes, or give me an X if you want to mix it up. Give me a yes if you're happy with what you had. And the first, 3 people to vote make the decision. Yeah, okay, basically yes. Okay, so we'll just go back there, 3 to 5, maybe 3. So why do blue-green algae dominate eutrophic and hypereutrophic lakes?
It looks like they create toxins.
Yeah.
Called like cyanotoxins. So apparently those are natural, powerful poisons. And it can make people, their pets, and animals sick.
Yeah.
I'm just wondering if that toxin would also make it so other, like, Other
other
algae.
plankton.
Yeah.
Yeah, I'm wondering that too. Could be an option.
It's the best one I've got so far.
I'm not quite sure, but maybe like since it could, let's see, it has like binary fission, so it could be like rapid reproduction as
Yeah.
well.
Well, because they create blooms, right? You've got algal blooms.
Yeah.
So it just seems like once they hit a certain amount of nutrients, they just soak that up and explode, basically. So maybe they like outgrow everything else at a high rate.
Yeah, that could probably be it
What
too.
was
Yeah.
what was the first thing that you said again?
That they produce by binary fission.
Okay. Let's do that. Binary fission.
Mm-hmm.
Okay. Oh, okay. But the toxins are produced when the bloom dies.
Okay.
So I don't know what that would have. Like what kind of effects that would. Obviously, if the bloom is dying, it's not doing too hot. So
Yeah.
that wouldn't help it grow in the first place. That wouldn't help it create a bloom. So
Could like maybe the nitrogen fixation also help?
yeah.
Yeah, okay. I'll see you then.
All right, welcome back. I think we got everybody back. Okay, I would say, just to save time, I'll tell you what I think. I think there are two reasons why blue-green algae are often, like very reliably, the most abundant type of algae in eutrophic and hypereutrophic lakes. And the first one is this. If a lake is very eutrophic, what that means is there's a lot of algae in the water, right? Lots and lots of algae in the water. You can see this. I have lots of algae here. It should be dots, but I'm saving time. And what that means is that if you're going to look at the light profile in this lake, this light, it's going to decrease really rapidly with depth because there's so much algae in the water. If you're something like a diatom, you're constantly falling, depending on water to move you back up into the light. If diatoms sink a meter a day, which kind of ballpark might be what they sink on average, the diatom, any individual diatom is going to spend most of its time in the dark below the compensation depth. So, if this is the compensation depth, 1%, in a eutrophic lake, it might be just within a meter of the surface. If the Secchi disk depth is 1 foot, then the compensation depth is 2 feet, approximately, roughly. Blue-green algae can control their buoyancy very effectively, very efficiently, right? If they do a lot of photosynthesis, they consume the CO2, the gas, and then they'll sink. If they don't do a lot of photosynthesis, they put more gas into the vacuoles and then they rise up. So they're actually able to get up into the light and maintain, without spending any energy, maintain the proper depth in the lake. Now you can do that if you have a flagella, but it takes energy. So I think a number one reason is that their ability to very efficiently control their buoyancy enables them to be very effective light competitors. If light is the limiting resource, they are a good competitor for the light. And this is competition in the ecology sense of competition. So in eutrophic lakes, the compensation depth is generally very close to the surface of the lake. And so you need to be like above the competition to get the light. So that's a big reason, I think, why cyanobacteria are so reliably a dominant alga, the dominant phytoplankton in eutrophic and especially hypereutrophic lakes, where the Secchi disk depth is really shallow, like a foot or 2. The second reason is that the reason an awful lot of lakes are eutrophic is because of humans. And what do humans do? We eat stuff, we create sewage. We just do, it's what we do. We turn food into sewage. Sewage is very high in phosphorus. This causes lakes to be enriched with phosphorus. Phosphorus is often the limiting nutrient, but if you add a lot of sewage to it, like even processed sewage, so it's coming out of a sewage treatment plant, it's been treated, it's still rich in phosphorus. The viruses are gone, the bacteria are gone, but the phosphorus is left. If there's a lot of phosphorus in a lake, which is not natural typically, but is the result of human activity, if there's a lot of phosphorus in a lake, then nitrogen becomes limiting. Well, if nitrogen is limiting, why would blue-green algae? Why would that benefit blue-green algae for nitrogen to be limiting? Anybody?
Because they can fix nitrogen.
Because they can fix nitrogen, right? If all of the other algae are limited by lack of nitrogen, because they've got all the phosphorus they need, so the thing that's limiting them is getting nitrogen. Blue-green algae can just pull it out of the air. Nobody else can do that. Nobody else can do that. The only other things on Earth that fix nitrogen are nitrogen-fixing soil bacteria and organisms that have cyanobacteria or nitrogen-fixing soil bacteria as endosymbionts. Like it's a really rare talent to fix nitrogen. Okay, so I think those are 2 of the main reasons why we see cyanobacteria so reliably dominating eutrophic lakes. And yeah, nitrogen is an essential element, right? You can't make protein without nitrogen. You can't make DNA without nitrogen. If you don't have protein, you can't make enzymes. You can't make enzymes, you can't live. No cell on Earth can live without nitrogen. It's an absolutely essential element. Phosphorus is also essential too for phospholipid membranes and DNA and RNA. But you need about 16 times as much nitrogen as phosphorus. In fact, you do at some point in this class need to know the Redfield ratio, which I just mentioned. And that is to say that phytoplankton need, and this is true not just of phytoplankton, but the Redfield ratio is about phytoplankton in particular. Phytoplankton need carbon, phytoplankton need nitrogen, phytoplankton need phosphorus, and they need them in about this kind of ratio, 106 to 16 to 1. You can't live without phosphorus, but you need 16 times as much nitrogen as you need phosphorus. And you need about over 100 times as much carbon as you need phosphorus. And this is called the Redfield ratio. So lakes are typically short of phosphorus. Phosphorus is typically present in less than a 16 to 1 ratio compared to nitrogen. And that means phosphorus is often the limiting nutrient in lakes. But humans put a lot of phosphorus in lakes because our sewage is quite, and our fertilizer is quite enriched in phosphorus. Yes, Ju.
Yeah, so I think I'm just kind of confused on the phosphorus aspect on eutrophic lakes and how and why the population of blue-greens would be more. So I mean, I get that the more eutrophic the lake is, that's because there's more phosphorus in the lake, but how does that increase the size of blue-greens population-wise?
When there is a lot of phosphorus into a lake, which is kind of unnatural, if there's an unnaturally high concentration of phosphorus, that means that phosphorus is not, that creates a situation in which phosphorus is not the limiting nutrient. And instead, next up the list, nitrogen becomes the limiting nutrient. So one reason that many lakes on Earth today are eutrophic is because they have been enriched with phosphorus by human activity. And in these lakes that are unnaturally enriched with phosphorus, nitrogen is the limiting nutrient because there's more than enough phosphorus. So that means nitrogen is what is limiting. And if nitrogen is limiting, then anybody who can get nitrogen is going to be able to reproduce. The blue-green algae can, some of them can get nitrogen just right out of the air and fix it. Does that answer?
Yeah, so like other algae, they need nitrogen as well too, right? But blue-greens are able to fix it, and that's why they dominate other algae.
Yep.
Okay.
Yeah, it's because they have special access to the limiting resource. We will talk more about nitrogen and phosphorus in the next unit when we get to water physics and water chemistry. But I think you can see we've already seen phosphorus on the x-axis of diagrams about algae. Obviously, it's very important for phytoplankton. Okay, let's move on. Somehow, the PowerPoint has extra animations in it. Okay, so let's go, let's change topics now, talk about phytoplankton size. You'll remember that we can talk about, we can give names to these size classes if you want to. People define them, different limnologists define them a little bit differently. You often see 2 as kind of it's a logarithmic scale using 2s. Okay, that's not really clear. What I'm saying is the teeny tiniest, the femto, this is like viruses. These are things that are less than 2 micron, 0.2 microns. Then you get to like, now why would we even include viruses on the list of phytoplankton? Because they're not photosynthetic, they're just viruses. Well, they can be absorbed Viruses can be absorbed and consumed heterotrophically. Even when a virus contains carbon and hydrogen and nitrogen and stuff, right? And phosphorus. Viruses contain important elements. And if you were to just neutralize the virus and digest it, you could use it as a source of elements, and you could actually use it as a source of energy. If you could respire it, right? So that's why it gets included on this list, I think, even though it's not photosynthetic. Then you got your picoplankton. That's maybe your smallest category of actual photosynthetic things. These are tiny, the tiniest of the cyanobacteria. Tiny little unicellular, tiny little unicellular whatever, cyanobacteria. Then you get a more typical size. These would be the nanoplankton. These are small. Okay, these are really small, 2 to 20 microns. But there are diatoms that are 10 microns in diameter. So now you're getting up into the small groups of normal algae. Then you get the micro, and these are things you can see with a microscope. You want to see algae with a light microscope? You're going to be looking at the microplankton. And there's, looking at it with the microscope, there's big things and there's small things, even on, a 100x lens microscope slide. And the big things are like the big microplankton, and the small things are the small microplankton. And the big and the small, they're all within this range. If you're looking at an algae with a light microscope, you're really looking at stuff between 2 and 200 microns. And 200 microns is 0.2 of a millimeter. If you start getting up into millimeter size, even close to 1 millimeter, even half of a millimeter, you can see that with your naked eye. You could see it in a jar. If there were little things in here that were half a micron in size, half a millimeter in size, you could see them. So maybe you wouldn't even use a regular compound microscope anymore. You might shift up to using like a dissecting scope to see them. You're in a different size category here. And that size category is called the mesoplankton. And once you get up above 2 millimeters, now you're talking, it's as big as a Daphnia. And that's macro, right? If the algae is as big as the Daphnia, the Daphnia are not going to eat it. So that's just a whole different thing. And that, in terms of, how it interacts with the world. And then it becomes called macroplankton. So these are the size categories. And, I don't know exactly how these were all named, but I kind of think the microplankton, well, this is what works for me. I think the microplankton are what you see with a light microscope. And then the names go the same way that the SI prefixes go. Milli, micro, nano, pico, femto. Millimeter, micrometer, nanometer, picometer, femtometer. So milli, micro, nano, pico, femto. So I think that those names are really just kind of borrowed from the SI, the metric system prefixes. And then when you go up from microplankton, micro means small, meso means medium, and macro means big. So I think that's how I think of, it's how I remember these names. And then just, if you just remember that, every category begins with a 2, the number 2, and then they go up by 10, by a factor of 10 each time. Within the microplankton, we kind of divide them into, things that are smaller than the typical mesh of a net, and things that are bigger than the typical mesh of a net, zooplankton net. And that's the divider within here. Not coincidentally, but I think there's a reason for this. This is the reason we use this size of the net. Things that are kind of in this size and above are not edible by zooplankton because they're the size of zooplankton. things that are kind of this size and smaller can be eaten by zooplankton, even the small zooplankton like rotifers. Okay, so this is a review of the phytoplankton size categories and some comments on why these sizes, they're more than just measurements. They're actually affect how the algae interact with other organisms, whether they're big enough to be eaten or not. And it also affects how they interact with the humans. Do we catch them or not? How do we look at them? Do we look at them with a light microscope, an electron microscope, or the naked eye, or a dissecting scope, or a magnifying glass? Okay, these are the size classes. Now let's look at the ecology that connects to size classes. Well, let's look at the big picture. Now you remember it goes milli, micro, nano, pico, right? So micro phytoplankton are bigger than nano phytoplankton, which are bigger than pico or pico phytoplankton. Okay, so these are three size classes. In this diagram, these are the biggest ones, these are the smallest ones, and these are in between. And what you see is this, a very strong pattern that where the total phosphorus concentration is high, so eutrophic, where in eutrophic lakes, they tend to be dominated by bigger algae. Where do you go if you want to see tiny, tiny little picoplankton? Where do you go if you want to see the tiniest ones? You go to oligotrophic lakes. Now, why is that? I think there's two big reasons why this is true, and one of them I've already mentioned. And the other one, I'm sure you have been taught one way or another at some point, probably in the cell biology part of General Biology 1. Okay, I'm gonna do a little foreshadowing, leave that in your mind to think about. And let's go to another size effect and ecology kind of slide. Better drink this coffee. Slip of the tongue here. Okay. Here again, here's another diagram that kind of shows the same pattern from a different data set and kind of in a different way. What we've got on the x-axis is total phosphorus. Obviously oligotrophic on the left, low phosphorus. Eutrophic on the right, high phosphorus. What we've got on the vertical axis is the pico phytoplankton biomass percentage. What percentage of the phytoplankton is picoplankton? Now you will recall that picoplankton is smaller than nano or micro, right? Picoplankton is smaller than micro or nano. And these are all your basically, your phytoplankton all fits in these three categories, unless it's like really big colonial stuff. Okay, so picoplankton is the smallest of the phytoplankton. Any smaller than that, you're really just talking viruses, like not entire cells. Where do you go if you want to see picoplankton? Oligotrophic lakes. In very oligotrophic, down here in the low single digits of total phosphorus, you see lots of these lake samples and ocean samples, or ocean samples. Lots of these were 75 to 100% picoplankton. Where do you go if you want to see bigger phytoplankton, nano, micro? Go to eutrophic lakes. Very strong pattern here. Now the x-axis is logarithmic and the y-axis is linear. So, what would this look like if this axis was linear? Okay, here's a cartoon of it. Professor helpfully includes a little cartoon here. You can see that the correlation is really quite strong. Really, I mean, a lot of these points are near the line. I mean, the difference between these points and these points here is largely explained by total phosphorus. That's what the R squared indicates. How much of the variation among lakes is explained by the x-axis variable? And here we've got an R squared of 0.89. And 0.89, this is R squared, so if you unsquared it, the R statistic, unsquared. The R statistic on this would be like 94%. But you always do it squared, so it's R squared. Sample size 87. What's the formula for this line? The percent picoplankton, that's our Y variable, is equal to 100. That's our y-intercept minus, because this is a downward slope, 37.2, that's the slope, negative 37.2, times x, and x is log of total phosphorus, because it's a logarithmic graph. So this is the equation for this line on this graph. Yeah, so when you see a graph with an equation on it, I strongly recommend that you look at the equation, look at the equation and figure out how that describes the line on the graph. Read the x-axis, what does it mean? Read the y-axis, what does it mean? What is each dot? In this case, the solid dots are lakes and the open circles are ocean or sea samples. What does each dot represent? And then what is the line? And there's the formula for the line. Here's the sample size. There should be 87 circles on this graph. It's the R squared. Okay, so hopefully, yes, okay. Any questions about this graph? Big picture, you know, it's kind of showing the same information, right? Low total phosphorus, greater dominance by the tiniest of algae. More total phosphorus, less dominance by the tiniest of algae. Very strong effect. Yeah, what could cause it? Well, here's another interesting correlation. The more Daphnia, and this was one particular lake, actually reservoir in Germany. And I think each dot represents a water sample from a different year, or the average for an entire year of water samples, which might be just the summer. I'm not sure if it was like full year round or just the summer season. Anyways, every dot is for a year. And what you see is that years in which there were more Daphnia, there was a lower concentration of nanoplankton. Now, nano is one step up from pico, but it's still small. It's smaller than microplankton. So years in which there's a high biomass of Daphnia are years of a low biomass of nanoplankton. Years of lower, so to the left is lower Daphnia concentration. Years of lower Daphnia concentration were years of higher nanoplankton concentration. Okay, that's enough about that diagram. And there's a nice little Daphnia picture for you, in case you forgot what Daphnia was. Gotta know Daphnia. Yeah, Daphnia. Okay, now you'll remember that some size classes of algae are considered edible. That's not edible to us, it's edible to the zooplankton, right? So we go back to our table. Now you remember that some size classes of phytoplankton are considered edible. They're called edible, and that means edible by zooplankton. What are those size classes? Picoplankton, nanoplankton, and the small microplankton. anything above about net size, about 70, maybe even a little less than that, is not really edible by the zooplankton. So we say edible plankton, it's those sort of 2.5 small size classes. Okay. So what we have in this graph is yet again on the x-axis, we have total phosphorus. We've got oligotrophic on the left and eutrophic on the right. And what you see is that as you get to higher, more eutrophic lakes, the biomass of the edible, just the mass, the density, grams per cubic meter of water, of the edible size classes increases. But it's not a very steep slope, right? In contrast, we can look at the productivity of the nanoplankton. And what you see is that in more eutrophic lakes, the nanoplankton are more productive. The nanoplankton are doing more photosynthesis, they're producing more biomass, they're producing more nanoplankton. And that this is a steeper slope than this one. Now, it may be a little hard to compare visually because these axes are both logarithmic, right? This is logarithmic, that's logarithmic, so it's maybe a little hard to think, well, what's the real shape of this relationship? So here's a helpful little cartoon here. And what this is showing is that as the total phosphorus increases, at first, there's a big increase in the biomass of this, of the edible size algae. But once you start getting into higher and higher phosphorus concentrations, it really kind of levels off. If you can, hopefully you can see that it's kind of a saturating curve. It's sort of rising rapidly first and then sort of almost leveling off. But the productivity of that size class, the activity of the photosynthesis of that size class is the opposite kind of behavior. As the phosphorus level in the lake rises higher and higher, as the lake, in a more and more eutrophic lake, that size class is more and more increasingly productive. So this is a little bit odd. How could they be more productive and yet there not really be that many more of them? How could their productivity increase rapidly while the number of them doesn't really increase rapidly? You can see, this is figure 2115 from the textbook. You can read this at your leisure a little more calmly. If you want to read it over again, read the sentence before it, read the sentence after it. It's described in the text a little bit too, as well, to give you some more description of it. But I hope I've given you a sense like, oh, this is really very odd. On the one hand, it makes sense, right? But on the other hand, it doesn't. Now, this is not percent of the biomass that is nanoplankton. This is the total mass of the nanoplankton. So in eutrophic lakes, there's more nanoplankton, but they are a smaller percentage of the total. In other words, other size classes benefit more than the nanoplankton do. In other words, like we've seen before, at at oligotrophic lakes is where you find lots of the smallest size classes. At more eutrophic lakes, that's where you find the bigger size classes, dominating, like the percent-wise. Okay, hope I've put that into words. Okay, so here's the question. What size of phytoplankton dominates in ultra-oligotrophic water like Crater Lake, Lake Tahoe, or if you want to think about oceans, the Antarctic Ocean, far from land? What size of phytoplankton do you think dominates? And now here's the question. Why? Okay, I'm gonna send you back to some version. I think actually I'm gonna send you to different breakout rooms, some new random ones, because I moved some people around and whatever. So that's the question. What size class of phytoplankton dominates ultra oligotrophic lakes and ultra oligotrophic open ocean, and why? And oh, that didn't work at all. Hold on. Let's just have 3 big rooms. See if you can come up with some answers. Oh my God. Is anyone here? Yes, you're still here. Okay. It just said no one was in a room. Okay, hold on a second here. Oh, I see. Well, you'd think I would know how to use Zoom at this point. Okay, here we go. 4 rooms, 4 people in a room. Come back with an answer. 3 minutes. Think about it. Brainstorm. What size class of algae dominates and why? 2 reasons, actually. All right, welcome back, everybody. I hope that you had some productive discussion in your group. And does anybody want to tell me what size class did you say? You want to just put it in the chat? Let's see what kind of, if we have a consensus here. Put it in the chat. What size class did your group think was going to dominate in oligotrophic lakes? All right, yep, we're getting the smallest size and everybody says pico. Yeah, and that's, of course, femto is smaller, but femto is really just like viruses and stuff. I mean, maybe there's something there, but yeah. All right, now why is that? Well, I mean, one reason is small particles fall very slowly. I don't know, that's maybe not a reason, that's just something important. You know, this is what I think the answer is, and I'll just tell it to you quickly. I think the answer that people usually say, limnologists and oceanographers, they say surface area to volume. If it's a very, very low nutrient environment, the rate at which algae take up nutrients is just all about as they drift through the water. So here's my algal cell. Jeffrey is an algal cell. Jeffrey's an algae cell. As Jeffrey drifts through the water, you know, at what rate does he take up nutrients? He doesn't eat or drink, he's just algae. Here, just cover your face there, pretend you're algae. Here, just like, okay. There. Yeah. So it's because the cell bumps into a dissolved nitrate ion, or it bumps into a dissolved phosphate ion. Right? Algae don't swim, they don't go out and grab nutrients. The only way they absorb nutrients is because the nutrients diffuse in from the water. And they do that at a rate that's proportional to the surface area. Because the, right? Does that make sense? I guess to me that's like so obvious, but I know that it could be kind of like, what? So it's all about surface area to volume. Let's say that we've got something that's I'm going to do a cube instead of a sphere because the formula for a cube is really easy. So let's say we have one algal cell that's 2 meters in diameter. It's a cube 2 by 2 by 2. The surface area, it has 6 faces. A cube has 6 faces. And every one of those faces has an area of 2 by 2, 4 square microns. The surface area of this cube is going to be 6, because there's 6 faces, times 4 square microns, because that's the area of one face. So the total surface area is 24 square microns. The volume of a cube is the length times the width times the height. In other words, 2 times 2 times 2, 2 times 2 times 2, cubic microns, 8. So the surface area to volume ratio is 24 to 8. And that's neither good nor bad to something bigger. Here, I've got a bigger cube, a big cell, 10 times as big, different size class, 20 by 20 by 20. What's the surface area? 6 times 20 times 20. 20 times 20, the surface area of this face is 20 by 20, so that's 400 square microns. It's got 6 of those faces, so the total surface area is 2,400. The volume of the cube is the length times the width times the height. 20 times 20 times 20, that's 248 times 10 times 10 times 10, 8 times 10 times 10 times 10. So this is the surface area to volume ratio of this. Okay, now there's a bunch of zeros in here, but what we had to start with was this one was 24 to 8 versus this one is, cross out some extra zeros to make it simpler looking, 24 to 80. This one has 10 times as much volume for every square centimeter of surface area. So does that make sense? So you went up, the dimension increased by 10, and the surface area to volume dropped by a factor of 10. Now, if there was plenty of nutrients in the water, there'd be lots of nutrients diffusing in from every direction, then it would be fine. But if the nutrients are really scarce in the water, then a smaller cell is going to be taking in more nutrients per unit volume all the time. It has an advantage. So anytime you're in an ultra oligotrophic lake, or even out in the open ocean far from land, because most of the nutrients wash into the ocean from terrestrial land, you're going to see tiny algae favored. And if I go, is that good? How many people got surface area to volume as one of your ideas for why the small ones dominate? Like when you were talking, did you think of surface area to volume? You could just give me a yes if that was one of the things that your group thought of, or give me a no if you guys had other ideas entirely. Surface area to volume, anybody? No? Oh, dang, well, I'm glad I explained that. I mean, because I thought that was so, I don't know, the first thing a biology student would think of, I get, frankly, because I don't know, it's so general biology. Okay. Glancing at the clock. Daylight savings time. Yeah. Yeah, and usually when they do the volume, usually when they do this in a class, they would do it with spheres. Because a cell is typically a sphere. But the formulas for area and volume of a sphere is not on the tip of anybody's tongue. It's just not, so it's not very intuitive. So that's why I used cubes. But hopefully you're like, oh, I think I get this now. Okay, I'm gonna go back to my slideshow and move along here. Okay, you ready for this one? You saw me mention this before. Here it is in more clearly, more simply, right? Phytoplankton biomass, and this is micrograms of carbon, which is a way to measure phytoplankton. Well, how much carbon is there per liter? So this is the density of them in water. Low concentration of phytoplankton in the water, high concentration of phytoplankton, oligotrophic on the left, eutrophic on the right. And on the vertical axis, productivity. Now this is micrograms of carbon produced per microgram of carbon per day. So if you had, 20 micrograms of carbon, oh, let's use one. Let's say you have 1 microgram of carbon. How many micrograms of carbon does it produce in a day? I mean, because it's not just carbon, it's algae, right? So you've got enough algae that you got a microgram of carbon. How much photosynthesis is it going to do in a day, right? That's what this vertical axis is. So this point here on the graph, for example, would mean, do I have a lake there? All right, let's pick this point here. Here's a lake, or here's a date on which there were 100 micrograms of carbon per liter of phytoplankton, 100, right? Because it's between the 0 and the 200. And it produced 50 micrograms per day in a day, almost half its body weight in new organic matter. Okay, so that's what each of these dots represent. And what you see is when there is more phytoplankton in the water, that phytoplankton is less productive. When you have less phytoplankton in the water, that phytoplankton is more productive. Here's the R squared. You can see there's a lot of scatter around the line. This line doesn't explain all of the variation. There's obviously other things going on in this data set. So the R squared's a little lower, but it's still pretty high. 67, that's pretty high. Here's the equation for the line. So this leads to this question. How can a eutrophic lake have more phytoplankton and yet be less productive? Or how could it have more phytoplankton and have that phytoplankton be less productive? It's kind of an economics question, isn't it, almost? And one of has less phytoplankton. And the answer to that is, oh, another picture. And the answer to that is zooplankton. You could have algae that's very productive, dividing every day, population doubling every day. But if you've got zooplankton that eat half the population every day, population's never going to grow. If you have a phytoplankton population that reproduces by cell division, which some of them do, wow, you could double every day. Just take in the nutrients and divide, take in the nutrients and divide, right? If you were a phytoplankton population and the cells divided every day, that population could double every day. But if there's a lot of Daphnia around, the Daphnia might eat a lot of phytoplankton. The Daphnia might eat half the phytoplankton every day. What do you got then? Huge productivity, no change in biomass. Okay, so that's one possibility, that you could have very high productivity and no change in biomass, effectively low biomass, because of herbivory. At the other end of the spectrum, you could have something like cyanobacteria that are basically maybe inedible. And you could have a huge biomass of cyanobacteria. If we go back to our diagram, you might have a huge biomass of cyanobacteria that are just sitting there. They're just hanging out. They're just doing enough photosynthesis to keep themselves alive. They're not producing any more cells. They're making sugar in the daytime, and then when the sun goes down, they burn the sugar. You know, they make enough sugar through photosynthesis just to support themselves. They're not supporting anybody else. They're not making any extra cells that get eaten by zooplankton. So you could in fact have very high biomass and very low productivity if it were something like blue-green algae that are inedible. On the other hand, you could have very low biomass and very high productivity, especially if it was something edible, like diatoms, or some of these pigmented flagellates. Small, very edible, very rapidly reproducing, but if they keep getting eaten. Okay, so it's not a paradox, but it requires you to know the difference between biomass and productivity, to know the relationship between biomass and productivity. Any questions about this question? Oh, I'm just going to check my chat here. Yeah. All right. Well, let's go to the final topic here, and I'm just going to talk about this 10 minutes. See how we go. What is the annual pattern? Okay, here's some great data from the North Basin of Lake Windermere. Lake Windermere is a lake in Great Britain in the Lake District of England, up in the northwest corner of England near Glasgow, kind of. It's a beautiful place. Lake Windermere is a very well-studied lake over the years. It's kind of beautiful, really. Anyways, we should go there. Anyways, there's a lake research station on the southwest side of the lake, and they've been doing research there for a long time. Anyways, very long data set. Very long data set here, 1964 to 1989. And what you see is quite consistently, in the summertime, the vertical axis is phytoplankton biomass, right? In the summertime, the biomass goes up, and in the winter it goes down. In the summer it goes up, in the winter it goes down. Summer goes up, in the winter it goes down. Summer goes up, right? Very, very consistent pattern. And also, if you look a little more closely, you see that the biomass goes up in the springtime, and then it drops a little bit, and then it goes back up again, and then it drops in the winter. It goes up in the springtime, it drops a little bit in early summer, and then goes back up again. Goes up, drops a little, goes back, goes down, up. So what we have here is a very consistent, pretty consistent pattern of a phytoplankton bloom in the springtime, and then a decrease in the phytoplankton, which is known as the clear water phase. The clear water phase, and then summer, and then down in the winter. Now, Lake Windermere is a warm monomictic lake. It stratifies in the summer, but it very, very rarely freezes over in the winter time. Basically, it's stratified in the summer, it mixes in the fall, it mixes all winter long and into the spring, and stratifies in the summer. So it mixes once a year, basically, you know, fall, winter, and spring, and it's stratified in the summer. But this is the same pattern we see in dimictic lakes, like in Minnesota. A spring bloom of algae, and then a clear water phase, and then a summer bloom that's typically higher, but not always. Typically, for us, it's always higher, but here, sometimes spring was even higher, more phytoplankton than summer. Okay. Now you remember we've talked about early in the semester, just the different types of mixing regimes, dimictic, amictic, monomictic, polymictic. Here's a diagram that kind of shows the relationship between them and their typical nature. This is just kind of a fun diagram if you like this kind of thing. Let's talk about dimictic lakes. Okay, dimictic lake has this basic pattern. It's ice covered in the winter, mixes in the spring, stratified in the summer, mixes in the fall, ice covered in the winter. Underneath this, I've got the months of the year, January, February, March, April, May, and so on. Okay, so now I'm going to show you some data. Okay, it's not data. Okay, now I'm going to show you a model. What is the classic dimictic, mesotrophic pattern, the classic pattern for lakes in Minnesota? For example, here's the pattern. In the springtime, shortly before the ice goes out, the diatoms start reproducing. They start dividing. There's a variety of reasons for this. But typically by the time the ice leaves the lake, the diatom population is already growing exponentially. The ice leaves the lake, the water mixes turbulently, there's lots of nutrients brought up from the bottom. The water movement helps suspend the diatoms, keep them suspended. And then if the ice or the snow on the ice was blocking light, now you've got more light. So you get a diatom bloom in the spring, partly because partly because the ice leaves the lake, and so there's more light, and there's more wind, increased water movement keeping the diatoms suspended, and also the increased wind mixing up more nutrients from the bottom water. Now this is all I'm going to say about it for now. Okay. Then the diatom population kind of crashes. And there's a clear water phase where the total amount of phytoplankton decreases. This is typically for us around early June. The water may be very clear. Then you get a bloom of green algae, and then the green algae are replaced by cyanobacteria. Typically in the summer, in mesotrophic and eutrophic lakes in the Twin Cities area, blue-green algae dominate in the summer. The whole time the lake is stratified. Lake starts mixing in the fall, stratification breaks down, and the lake starts mixing top to bottom. The blue-green algae tend to die out, and you often have a second fall bloom of diatoms. So the brown or golden colored line here is showing, am I trying to draw? I do this better when I draw it on the whiteboard in a classroom, but anyways, I tried to draw it on PowerPoint. And what we got is a spring diatom bloom, followed by a clear water phase, followed by a green algae bloom, which is then replaced by a blue-green algae bloom, right? All this green algae makes the water murky. This favors the blue-green algae, which can float above the competition, right? And get the light, because often light is limiting. But then when the lake starts to mix again, the water gets cold, silica is brought up from the bottom water, the diatoms bloom again, and the blue-green algae die out, and then the ice and so on. Okay, so that's your typical annual pattern. Typical, classic. Now I will say, Dr. Shapiro used to say, every year is different, and every lake is unique. So, you know, it's not like, it's like always identical to this for all lakes every year. But we might consider, why is it that diatoms are often less abundant in May than in April? You know, what is it that causes this decrease? I think we'll return to this question on Monday. I want to look at like ecology here, right? Do we really see this pattern, this classic pattern? What does it look like in some of these well-studied lakes in other parts of the world? Okay, here's Lake Erkin. Here you see the ice in the winter, stratified in the summer, mixing before and after. So classic dimictic lake. Here's the total phytoplankton biomass. That's the solid line. You see it rises in April and May after the ice goes out. So here's the spring diatom bloom. And then it drops, that's the clear water phase. And then it rises again in late summer to an even higher peak. And then drops back down again. Maybe there's another peak in the fall. It's not nearly as pronounced as the peak in the spring. That's the solid line. The dotted line is productivity. And you can see that here in late summer, the phytoplankton productivity is not really all that much higher than it is earlier in the summer. And yet the biomass of the algae is much, much greater. Like there's twice as much phytoplankton in August as there was in whatever, late June. Twice as much, much more than twice as much phytoplankton in the water. And yet the productivity is only slightly higher. They also have colored in the microplankton, right? So there's more information in here. Who are these algae? Well, let's look at this summer bloom in particular. This great big peak is like 75, 80%. Oh, dinoflagellates. That's really weird, isn't it? Now the top layer here that's not colored at all is the blue-green algae. And you can see the blue-green algae are just a thin percentage of the total. But then here in this lake in July, you got the phytoplankton dominated by blue-green algae. How about those diatoms, the Bacillariophyta? The Bacillariophyta are this kind of color right in the middle. It's kind of gray right in the middle. And you can see that they really dominate the phytoplankton in the winter, even though there's not a lot of phytoplankton there. And then this spring bloom, this high amount of total biomass is dominated by diatoms. And then again in the fall, you sort of got a smaller peak of biomass here in the fall and into the winter, and it is again dominated by diatoms. So this is some real data, and there's more information in this diagram than just these cartoon lines here. But I think you can see that a lot of this pattern is repeated, is also shown in this information from Lake Erkin. In this PowerPoint, which is posted, which we'll finish next week, I also want to raise the question, what's the role of herbivory in creating this pattern? How does this pattern look in completely different lakes, like Tanganyika, which is an important lake, but is warm and monomictic? Or Char Lake, which is a well-studied ultra-oligotrophic boreal lake. And then like, what about a polymictic reservoir, a shallow reservoir that mixes multiple times? And that, we'll talk about that later. It's supplemental information. So we'll, on Tuesday of next week, we will finish talking about the annual cycle of phytoplankton and use that to review things about the phytoplankton and the zooplankton and the fish. We will discuss the paper by Shapiro and Wright that is posted. Be sure to read that paper. You don't have to understand every word, but attempt every paragraph. Go through the article and try to read every paragraph. If you come to a paragraph that you can't understand, go to the next one and look at the figures. And if you have questions as you are reviewing, put them in the survey on D2L. That's what I want to say about Tuesday of next week, which is our next lecture meeting. Thursday of this week, we're going to meet at Lake Phelan and Roundway Lake to look at the ice. We will probably not go on the ice. If it looks okay, and if anybody wants to, we'll send you out to measure the ice. I got one auger and a rope, but don't worry. If the ice is bad, you don't have to go out on it, right? But Bree's gonna want to. Some people, is she there? If you don't like the lab groups that I created here, then send me an email. There's still some room to shift around a little bit. Otherwise, I expect to see Abdul, James, Kat, and Ali, Bree, and Jang at 1 p.m. Hannah, Desiree, Christina, Brett, and Tiffany at 2 p.m. Keenan, Jew, Sam, and Saul. I hope we see Saul at 3 p.m. on Thursday. Okay, I'm going to stop sharing this slide. Let's see if we have questions. Bree says, send me out there. You see, I knew it. All right, any questions? Anybody? All good? You can reach me by email. I was totally offline all spring break, but now I'm back. So, questions? Class ends at 4, right? I'm not making that up. Yeah. No, daylight savings time. It doesn't save light, does it? It just saves electricity. They should call it electricity. Bye, Kat.